covering nervous tissue. So let's go ahead and begin. The nervous system is a master controlling and communicating system of body. Cells communicate via electrical and chemical signals. It's a rapid and specific communication, usually causes almost immediate response. Now, when we're talking about communication between cells via electrical signals, what we mean, electrical impulse called action potential, and chemical signals are neurotransmitters. But important information to remember that it's a very rapid, specific, and immediate response. Now, nervous processing include sensory input, information gathered by sensory receptors about internal and external changes. Like on a diagram, you can see sensory input over here through visual input. So you see the glass of water. So that's a sensory information received by your special organ, eye, and electrical impulse sent to the brain. Then we have integration, interpretation of sensory input that happen in your brain. You have connection with what is called interneuron over here, and motor output, activation of effector organs. Effector organs include muscles and glands that produces response. So you see motor output to skeletal muscle, and it allows you to pick up the glass. So again, sensory input, integration, and motor output. Central nervous system, abbreviation CNS, includes brain and spinal cord, integration and command center. Peripheral nervous system, PNS, includes paired spinal and cranial nerves that carry messages to and from the CNS. Also part of the peripheral nervous system, all the rest of nervous tissue found in your body. For example, different receptors that found in your skin, receptors found in your muscles, they're part of the peripheral nervous system. So here on the diagram, you can see that nervous system divided into central nervous system, brain, spinal cord, and peripheral nervous system. Major organs are cranial nerve and spinal nerves, and all nervous tissue outside of the central nervous system. Now, if we look at division of the peripheral nervous system, it includes sensory or afferent, start with A, afferent division, somatic and visceral sensory nerve fibers. Now, somatic nerve fibers comes from your skin and your muscles, and they reach your consciousness. Visceral nerve fibers come from your internal organ. They conduct impulses from receptors to the CNS. And motor output or afferent start with E, afferent division. So afferent and afferent. Motor nerve fibers that conduct impulses from the CNS to effectors, muscles, and glands. And afferent division include somatic nervous system, somatic motor neurons that are voluntary, conducts impulses from the CNS to skeletal muscles, and autonomic nervous system, ANS, visceral motor involuntary neurons, conduct impulses from the CNS to cardiac muscles, smooth muscles, and glands. ANS is divided into sympathetic division, mobilizes body system during activities. This is what is called fight or flight response. And parasympathetic division conserves energy. promotes housekeeping functions during rest. This is called rest and digest system. And also peripheral nervous system has input to central nervous system for integration and control, and a nervous system sends output to peripheral nervous system. PNS, the functional division, that's what we already explained using the diagram. So now we're going to repeat it. So peripheral nervous system that mostly includes your nerves has a sensory or afferent division. Somatic afferent fibers conveys impulses from skin, skeletal muscles, and joints. So sensory information from your skin, your muscle, and your joints moves through this afferent neurons. And visceral afferent neurons convey impulses from visceral organs. And motor or afferent division transmits impulses from the CNS to effector organs. Now, what are those effector organs? Those effector organs include somatic, well, if it's somatic nervous system, then your effector organs are your skeletal muscles. So motor division of PNS includes somatic or voluntary nervous system, conscious control of skeletal muscles, and autonomic, involuntary nervous system, ANS, visceral motor nerve fibers that regulate smooth muscles, cardiac muscles, and glands. And ANS has two functional divisions: sympathetic, fight or flight response, and parasympathetic, rest and digest response, right? So, so just to look at a big picture, you have brain, right, and spinal cord. That's your central nervous system. And then you have nerves, and nerves carry information from your periphery, from your skin, from your muscles, from your internal organs. That information is carried from periphery to your central nervous system is called sensory information. And if it's carried from your skin, your muscles, and your joints, then it's somatic sensory information. If information is carried from your visceral organs, it's a visceral sensory information. Then your brain decides what to do with this information, how to respond, and it's gonna send signals to your effector organs, organs that can do something. What organs can do stuff? Those your skeletal muscles, they can contract and you can move parts of your body. Or your smooth muscles, they can contract, like muscles of your intestine, of your stomach, muscle of your urinary bladder, or even muscle of your blood vessels. And muscles of your heart also can contract, they also effector organs, and also glands. Glands can secrete. So if your brain sends information to your skeletal muscles, then you call it somatic neurons, and it's your somatic nervous system. If your brain sends information to your internal organs, you call it autonomic nervous system, or ANS. And autonomic nervous system divided into fight or flight, or sympathetic, and rest and digest, or parasympathetic. Right, so that's like on this diagram, we pretty much can see the same information, how your central nervous system communicate with the peripheral nervous system, and also shows you somatic sensory fibers and visceral sensory fibers, and motor fibers of somatic nervous system to your skeletal muscles, and motor fibers of your autonomic nervous system. Sympathetic motor fibers of ANS and parasympathetic motor fibers of ANS, autonomic nervous system. Okay, now that was overview of the nervous system. Now let's go ahead and discuss nervous tissue. Histology of nervous tissue. Two principal cells, neurons, excitable cells that transmit electrical signals. So on this diagram, neurons are shown in yellow color. So here's a neuron with long processes. And second type of cells, neuroglia or glial cells, supporting cells, including astrocytes, microglia, ependymal cells, oligodendrocytes. Those are cells in the central nervous system. And then two S's, satellite cells and Schwann cells found in the peripheral nervous system. CNS astrocytes, most abundant, versatile, and highly branched glial cells, cling to neurons, synaptic endings, and capillaries, support and brace neurons, help determine capillary permeability, control the chemical environment, participate in information processing in the brain. So you can see those cells, astrocytes. Astro means star, so they shape of a star. They have pseudopods, like little feet. And look what those feet do. They cover blood vessels in your central nervous system. And also some of these feet attach to neuronal processes. But when they cover blood vessels, they control capillary permeability. So they are important in creating blood-brain barrier. Because if you have, for example, some bacteria inside the blood, you don't want this bacteria to easily move out and infect your brain tissue. So astrocytes, very important to control what can move in and out of blood vessels inside central nervous system. Also, they support, protect, form the network, that supporting network for neurons, a control chemical environment around neurons. Microglia, small ovoid cells with horny processes, migrate toward injured neurons, phagocytize microorganisms and neuronal debris. So microglia are small cells, but very important in the protection of neurons. So they like almost like a little like you have phagocytes, so they have phagocytic properties as well. So they can destroy bacteria, other pathogens, and also, well, they do not help to neurons to regenerate, because neurons in the central nervous system cannot regenerate, but they can help to get rid of some debris or pathogens. Ependymal cells range in shape from squamous to columnar, may be ciliated, line the central cavities of the brain and spinal column, separate the CNS interstitial fluid from cerebrospinal fluid in the cavities. So ependymal cells line cerebrospinal fluid field cavity. So on this diagram, you can see brain tissue, ependymal cells, and fluid field cavity. And those cilia, when it beats, it's also allow the fluid, cerebrospinal fluid, to move. Oligodendrocytes are branched cells, processes wrap CNS nerve fibers, forming insulating myelin sheath, equivalent to the Schwann cells in the PNS. So this yellow structure that you see, those are dendrites, processes of neurons, and that's an oligodendrocyte that produce this covering around nerve fibers or dendrites and axons, mostly around axons, actually. So it's produced this myelin sheath around axons, and it helps to insulate axons and increase speed of electrical conduction. Also interesting that one cell can wrap several axons. So here we have several nerve fibers, several axons, and one cell forming this myelin protection around several nerve fibers. Now, neuroglia of peripheral nervous system include satellite cells, surround neuron cell bodies in PNS, and they're similar to astrocytes. So satellite cells and astrocytes, they have similar functions. The difference is astrocytes found in central nervous system and satellite cells in the peripheral nervous system. And of course, we have other differences, but here you can see satellite cells that kind of cover the cell body of a neuron, and it's protect, nourish, regulate, again, chemical environment around neurons. And Schwann cells, another name is neurolimocytes. They're similar to oligodendrocytes because they also form myelin sheath, surround peripheral nerve fibers and form myelin sheath. So that's our myelin sheath. And myelin sheath is like chemical that is made of mostly made of lipids and proteins. So it's lipids and proteins that cover axons of neuron. They also, Schwann cells are vital to regeneration of damaged peripheral nerve fibers. So it's interesting that Schwann cells, they help your nerves to regenerate if they damage. However, oligodendrocytes, they do not have this function. They actually even secrete some chemicals that prevent neurons of the central nervous system from regeneration. So what I'm trying to say here, that your nerve fibers in peripheral nervous system, your nerves, they can regenerate. So if you have a cut, if you broke your leg or arm, those nerves might grow back. But if there is a damage of spinal cord or brain, those neurons cannot regenerate. So those connections, those axons, if they are cut and damaged, you don't regrow them again. And now let's talk about neurons. Special characteristic, long-lived, 100 years or more. So they live as long as we do. Amitotic, that means they don't divide, with few exceptions. High metabolic rate depends on continuous supply of oxygen and glucose. Now, why do you need oxygen and glucose? To make ATP. And ATP is needed for metabolic reactions. Plasma membrane function in electrical signaling. So here you can see a structure of a neuron. It includes the body of neuron or soma. Inside you will find nucleus, nucleolus, then lots of other organelles like mitochondria, micelle bodies. Those are organelles that very specific to neurons. Then you have shorter processes, highly branched, called dendrites, and one long process called axon. Now, the area where axon branches from the body is called axon hillock. At the end of axon, you will find axon terminal. That's a secretory region where neuron secretes neurotransmitters. You will see terminal branches of axon, and this yellow, this blue part shown over here, that's a myelin sheath made by Schwann's cells. Now you can see it's not continuous covering. We have gaps between myelin, and those gaps are called node of Ranvier. And that shows you a Schwann cell. So cell body, or Soma, biosynthetic center of a neuron, spherical nucleus with nucleolus, well-developed Golgi apparatus, rough ER called missile bodies, axon hillock, cone-shaped area from which axon arises, and clusters of cell bodies are called nuclei, nuclei in the central nervous system and ganglia in the peripheral nervous system. Let me very quickly explain what this the last sentence means. So if we draw axon, neuron, right? So here's our cell body with the nucleus. Here we have dendrites, right? So here lots of dendrites, and then we have axon. And if we put another next to it, cell body with the dendrites, right? So here's all dendrites and long axon. Now those cluster of these cell bodies, clusters of cell bodies, if it's in a central nervous system, we call this cluster nucleus. So do not confuse with this nucleus inside. Just a cluster of cell bodies called nucleus. If we have this cluster of cell bodies in the peripheral nervous system, we call it ganglia, right? So that's what it the last sentence means. Dendrites are short, tapering, and diffusely branched neuronal processes. They are receptive region of a neuron, so they get input. So information travels through the so for example, you have like a receptor maybe here, and this receptor gonna send information about temperature to a dendrite. Now this information travels along dendrites towards the cell body, so they are input regions or receptive regions. They convey electrical signals towards the cell body as graded potential. Now that's electrical signals that you send towards the cell body has a special name. It's called graded potential. Axon, one axon per cell arising from axon hillock. So here's our axon hillock, and it will be important in physiology because this is the most excitable part of neuron. Long axons, and we call those axons nerve fibers. Occasional branches called axon collateral, so as shown over here, for example. Numerous terminal branches, so we have lots of terminal branches called telodendria. Knob-like axon terminals, so you will have like knobs over here called synaptic knobs or buttons. They are secretory region of neuron, release neurotransmitters to excite or inhibit other cells. function of an axon. It's a conducting region of a neuron. It generates and transmits nerve impulses that called action potential away from the cell body. And here you can see dendrites collect electrical signals, send signals towards the cell body. Cell body integrates incoming signals and generates outgoing signal to axons, and axon passes electrical signals to dendrite of another cell or to an effector cell. Now, these electrical signals that dendrite cells to the body are called graded potential, and electrical signals that travel across axon, that's sent by axon, called action potential. Myelin sheath, segmented protein lipid sheets around most long or large diameter axons. It functions to protect and electrically insulate the axon, increase speed of nerve impulse transmission. So you can see on the diagram, this is neuron found in CNS. How do we know it? Because they look really, really similar, but you can see oligodendrocytes. It's oligodendrocytes that make myelin sheaths along several nerve fibers. And this is neuron in the peripheral nervous system because we have Schwann cells and they form myelin sheath along one nerve fiber. And both in central and peripheral nervous system, you see gaps between myelin called node of Ranvier. So Schwann cells wrap many times around the axon. Myelin sheaths, it's concentric layer of Schwann cell membrane, and neurolema is peripheral bulge of Schwann cell cytoplasm. So look how interesting is over here. So here's the axon. That's a cell. Schwann cell, it has a nucleus, right? It has a cell membrane. And now it wraps this membrane several times around these axons. Now nucleus and all the organelles stays in the outermost layer. Now this outermost layer of Schwann cells called neurolema, and the membrane of axon called axolema. Unmyelinated axons. Thin nerve fibers are unmyelinated. One Schwann cells may incompletely enclose 15 or more unmyelinated axons. So in the diagram, you can see Schwann cells that start to envelope multiple axons, and the unmyelinated axons are enveloped by the Schwann cells, but they are no myelin sheaths wraps around each axon. So, so not only we might have, let's say, nerve fibers that are unmyelinated, or they kind of like partially have myelin around them, but it doesn't cover entire axon. Nodes of Ranvier. Myelin sheath gaps between adjacent Schwann cells. Site where axon collaterals can emerge, right? So if you have a collateral, a branch, it will emerge from the node of Ranvier. Okay, so now let me remove this. Cannot remove it. Very interesting. Okay. Myelin sheaths of central nervous system formed by processes of oligodendrocytes, not the whole cell. Nodes of Ranvier are present, no neurolema, and CNS fibers are unmyelinated. Now, you probably heard about white matter and gray matter. So first thing I want to tell you, white matter and gray matter found only in the central nervous system. So not in your nerves, but in your brain and spinal cord. White matter is a dense collection of myelinated fibers. So those axons with the myelin, they appears lighter color. And the gray matter, mostly neuron cell bodies and unmyelinated fibers. So when you have myelin, it looks light or white. If you have a cell bodies or dendrites or unmyelinated axons, then it's gonna look darker, and you call it gray matter. So you see, this is your spinal cord. You would find gray matter inside and white matter surrounding gray matter. In the brain, you have gray matter that form cortex, so that's a cerebral cortex. You have also gray matter inside the white matter called nuclei, and this is white matter in between. So again, white matter are axons that is myelinated. Gray matter are cell bodies, dendrites, and unmyelinated axons. Processes, dendrites and axons. Now, bundles of processes, mostly axons, are called, in CNS, they called tract. In PNS, they call nerves. Right, so here you see nerves branching from the spinal cord. That means those are axons, right? And here you see tract in the central nervous system, so those are axons as well. But remember, let me again draw, because neurons, so here's your cell body with dendrites, and here's your axon, and here you have another neuron, then you have dendrites, and you have long axon. Now, this collection of cell bodies are called nucleus in CNS and ganglia in PNS. And those bundles of axons are called tract in CNS or nerves in PNS. Structural characteristic of neurons. 3 types: multipolar, one axon and several dendrites. So here's the cell body. We have lots of dendrites and one axon. Sorry, let me draw it for you. Okay, so here we have cell body. No, okay, it's not drawing. Oh, here. Okay, cell body, multiple dendrites, right? Multiple dendrites and one axon. If we have structure like that, one axon and several dendrites, we call it multipolar. Most abundant motor neurons and interneurons are multipolar. Bipolar, we have one axon and one dendrite. So how would you draw this? So here's your cell body, and you have one axon or dendrite and one axon. So they're rare. An example gonna be neurons of retina, and also your olfactory neurons that allow you to smell. In unipolar, actually it's like a pseudo-unipolar, single short process that has 2 branches. So that would look like this. So you have your cell body, right? And then you have one process only, and then it splits into 2. Right? Peripheral processes, a more distal branch, often associated with sensory receptors. So that's going to be sensory, go to periphery, goes to your skin, to your muscles, and central process, a branch entering the central nervous system. So we don't really call it a neuron or a dendrite or axon. We call it peripheral process, go to periphery of your body, and central process goes to central system. Here, if you didn't like my drawings, and I'm not gonna blame you for not liking it, then you can see drawings done by our publisher. So we have a cell body, multiple dendrite axons, and this is our multipolar. Here's our bipolar, 2 processes, and pseudo-unipolar. So many processes extend from the cell body. All are dendrites except for a single axon. And a receptive region, the one that receives stimulus, shown over here in blue, and plasma membrane exhibits chemically gated ion channels. So here's going to be our conducting zone, and this is going to be our secretory region, right? So receptive region, conductive region, and secretory region. If it's a bipolar neuron, two processes extend from the cell body. One is a fused dendrite, the other is an axon. Then here's our receptive zone. So here's our receptive zone, then conducting zone, and secretory region, or zone, region, you call it region. Then pseudo-unipolar, one process extends from the cell body and forms central and peripheral processes, which together comprise an axon. So here we have peripheral process and central process, and both kind of peripheral and central process considered to be axons. That's why our receptive zone is really small over here. Then we have conducting zone and secretory zone. Why I call it zone? It's region, but it's okay. I think you can say zone or region. And you can see many other neurons. They all kind of, well, for example, this Purkinje cells in cerebellum, but this is a multipolar neuron. Pyramidal cells, also multipolar neuron. Now you can see bipolar neurons, that olfactory cells, I told you, the ones that allow you to smell your surroundings, and neuron in the retina, retinal cells, those are bipolar, and unipolar. The example is a sensory, all sensory neurons are unipolar. This one is dorsal root ganglion cell. So it has central process, so that go to the brain, and this is a peripheral process where you receive sensory information. So let's read about this unipolar. Found mainly in a peripheral nervous system, common only in the dorsal root ganglia of the cranial, spinal cord and sensory ganglia of cranial nerves. Yeah, I didn't explain you what this dorsal root ganglia is, but we will look at this dorsal root ganglia where it's located in our central nervous system lecture. So then you can watch that lecture to understand more what this dorsal root ganglia is. Functional, so that was structural characteristic, right? Based on structure, on morphology, how many processes branch from cell body. We can also classify neurons by functions. And also three types: sensory or afferent, transmit impulses from sensory receptors towards the central nervous system; motor or afferent, carry impulses from central nervous system to effectors. Remember, our effectors: skeletal muscles, smooth muscles, cardiac muscles, and glands. And interneurons, association neurons, shuttle signals through central nervous system pathway. Most are entirely within CNS. This diagram shows you functional classification. So you see this lady skin, right? You do have receptors in your skin. So if somebody touches you, you can feel also sensation of cold and hot and pressure and vibration. Now in your skin, you will have your afferent neurons. So here's this afferent neuron. So you generate signal, electrical signals, and it's kind of like travels through this nerve, right? So by the way, you see that's your posterior root ganglion. this is unipolar neuron. You see one process, and then we have peripheral process and central process. So that's our peripheral process. Information travels towards the cell body, and then guess where? In the spinal cord, right? In the spinal cord, it's synapses with interneuron. It's entirely within spinal cord, within central nervous system. Now, this neuron sends information to motor neuron, right? So here's the shown in blue. That's a motor neuron, and it goes through the ventral root of the spinal nerve, right? And where it goes through the efferent pathway towards your muscles, and then you contract your muscles. So maybe you have like a little bug crawling on your skin, right? You send this information to your spinal cord, synapses with interneuron, with motor neuron, send information to your arm, and you shake, and you kind of like remove this bug, right? You shake it off. That's a functional characteristic of the neurons. But of course, keep in mind that it's not going to be just one single synapse over here, only with this interneuron. You also will send information to your brain, so you can understand, oh, look, somebody's crawling on me, or I feel it's cold or hot by my skin. Now, what is synapse? Synapse is a junction that mediates information transfer from one neuron to another neuron or to an effector cell. Now, here is our axon. This is axon terminal. And this axon terminal synapses with blood vessel. So this is effector cell. This neuron number 2, cannot see what it's connected. Oh, right there, that's still 2. It's synapses with dendrite of another neuron, right? So that's also synapse. So here we can have synapses with cell body, for example. So now synapse, oh, okay, I think we will talk about synapses more. Now look, there is no direct connection between axon. So look, this is axon and this is dendrite of another neuron. There is no direct connection. There is actually a space in between, right? Now the whole structure is called synapse. Now we have presynaptic neuron, shown here in blue, that conducts impulses towards the synapse, and postsynaptic neuron, shown in pink, that conducts impulses away from synapse. Now, what this presynaptic neuron does, it releases neurotransmitters and activates postsynaptic neuron. Now, we have 2 types of synapses. Electrical synapse, shown right here. Neurons are joined by gap junctions. So we do have space, right? But what join or connects 2 neurons are those channels called gap junctions. And there is no neurotransmitter over here. Look, there is no neurotransmitter. So electrical signals move through those channels from neuron to neuron. Communication is very rapid. It's less common, found in the heart. So for some reason, students always think because neurons send electrical impulses, we have electrical synapses. No, electrical synapses are not common. Then what type of synapse we have? Chemical synapse. Why do we call it chemical synapse? Because we release chemical within this gap between 2 cells. Those chemicals are called neurotransmitters. So chemical synapses are most common. They specialized for the release and reception of neurotransmitters. Synaptic cleft. It's that fluid filled space separating presynaptic. Okay, which one is presynaptic over here? Well, look, the one that secrete neurotransmitters. So separating presynaptic and postsynaptic neuron, and this gap is called synaptic cleft. Prevents nerve impulses from directly passing from one neuron to the next. Involves release, diffusion, and binding of neurotransmitters. So here's your release, Here's your diffusion, and the neurotransmitter binds to its receptor. And this is how you pass information from one neuron to another. So neurotransmitters is a chemical that is released from a nerve cell, which is thereby transmit an impulse from a nerve cell to another nerve, muscle, organ, or other tissue. We have 50 or more neurotransmitters that have been identified. Most neurons make 2 or more neurotransmitters. Well, here are some chemicals that are very familiar. Norepinephrine, dopamine, serotonin. They are released by neurons, so by definition, they are neurotransmitters. But not only they carry information from neuron to neuron, they also affect your mood, your anxiety, your attention, motivation, obsession, compulsion, energy, and many other functions. Why synapses are important? We know that synapses is when we transmit impulse from one neuron to another neuron. So synapses allow us to transmit that impulse. They control the direction of the impulse and only allow it to pass in one direction. Neurotransmitter receptors are only found on the postsynaptic side of synapse. So we prevent this electricity going back and forth in your body. We want impulse go in one direction. Now, impulse can be blocked by certain chemicals. So if you have a synapse and you look over here, and for example, you have a chemical that will block this receptor, that impulse cannot be transmitted. And this is what happens when you have some, for example, painkillers. It also prevents overstimulation of effectors, muscles or glands. Neurotransmitters stop being produced due to constant stimulation, such as pain or noise, for example. So if you have a lot of pain, constant pain of noise, then you start adjusting the amount of neurotransmitters that is released, and it's prevent overstimulation of your receptors. Now, action potential, propagation. Now, that electrical impulse that is carried by neurons and specifically by axons, right? So if you have this electrical impulse that carried by axon, we call it action potential. Now, it can be continuous conduction or saltatory conduction. Now, continuous conduction, when we have unmyelinated axons, and it's very slow. When we have saltatory conduction, the impulse it's kind of jumps from one node of Ranvier to another node of Ranvier. So if you want to move from point A to point B, and you just go slowly, or you jump from point A to point B, or then your, of course, speed of conduction is higher in saltatory conduction. Saltatory conduction in myelinated axons and action potential jumps from one node of Ranvier. Remember this area where we don't have myelin, so this is myelin. This point here and over here where we don't have myelin called node of Ranvier, and action potential jumps. And this is where you have channels that allow ions to move inside and outside of neuron. When it's continuous conduction, obviously conduction will be slower. Now, sometimes you're supposed to have saltatory conduction and myelinated axons, but then this myelin sheet is destroyed, and then impulse slows down and it can completely stop. And when this happens, that's disease. And we get to those conditions in a minute. Let's just compare again saltatory and continuous conduction. Saltatory conduction is the propagation of action potential. Keep in mind that's electrical impulse between the nodes of Ranvier in myelinated axons. Continuous conduction is the propagation of action potential or nerve impulse along the entire length of the unmyelinated axons. Transmission in saltatory between nodes of Ranvier in continuous along the entire length of axon. Speed, faster, saltatory, slow, continuous. Saltatory occurs in myelinated axons, continuous, unmyelinated axons. Efficiency, saltatory is more efficient, continuous, less efficient. And we're gonna look at the energy use of energy is low for saltatory and high in the continuous transmission. Now, what I said before, that sometimes a myelin sheath is destroyed. Now, why would it happen? One of the reasons might be because of your own immune system. When your immune system destroys a myelin sheath, the person said to have disease. Now, if it's happened in the central nervous system, it's called multiple sclerosis or MS. If it happened in the peripheral nervous system, it's called Guillain-Barré syndrome. Now, Guillain-Barré syndrome, GBS. So they both are similar, and at the same time, there is differences between them. Similarities are that myelin sheath is destroyed either in your brain or spinal cord, somebody's brain and spinal cord. Then it MS. If myelin sheath destroyed in the nerves, spinal nerves or cranial nerves, then it GBS. Autoimmune disease, when immune system attacks and damages myelin. Now, think about person with multiple sclerosis. If neurons in brain and spinal cord are affected, right, then the symptoms can be like really, they can vary, because your brain and spinal cord controls many functions. Now, all of these functions can be affected. Now, MS affects young adults. Symptoms, look how different symptoms over here. Visual disturbance, weakness, loss of muscular control, speech disturbance, urinary incontinence. And on a diagram, you can see even more different symptoms like fatigue, depression, anxiety, speech disturbance, then loss of muscle control. It's affect your sensation and pain and bowel movement and urinary system function. And again, why it happened? Because brain and spinal cord controls all of these functions. Now, myelin sheath in the CNS become non-functional sclerosis. Shunting and short circling of nerve impulses occurs, and impulse conduction slows and eventually ceases. Oops, and I think that's it. Sorry, I didn't realize that's my last PowerPoint slide. Okay, and maybe what I want to add that, of course, in a Guillain-Barré syndrome, we also have muscles that are affected and sensory functions that is altered, because your nerve and also can affect your internal organs, because you have nerves that innervate your skin, your muscles, and your viscera. So when those nerves affected by myelin damage, then the function that controlled by those nerves are also altered. Okay, but anyway, that's it for this PowerPoint. I'm just gonna remind you that we covered nervous tissue. Thank you for watching, and I hope it was helpful.